Okay, now we shall move on to mechanism of perception. Okay, we already see on how the colors are being perceived. Okay, how the colors is actually, and I mean how the lights enter to the eye, and then we able to perceive the colors. Okay, now we're going to look at how the retina is actually reacting to this lights that enters into the eye, and what are the components in the eye that is actually responsible for color perception. So first we have to know the human eye and photoreceptors. Okay. So photoreceptors called cones are sensitive to different wavelengths of light. Okay, there are three types of cones, each sensitive to red light, green light, and blue light. So basically this without photoreceptors, okay, the cones, okay, in the retina, we have a structure that is known to be cones, okay, which is actually very sensitive to this different wavelengths of light. So this cones, without these cones, then we won't be able to actually differentiate the wavelengths that enters to the eye. Okay, so these cones are, there are three types of cones in a human eye. Okay, one is sensitive to longer wavelength, which is longer wavelength lights, which is more to red shades. And then we have another type of cone, which is sensitive to medium wavelength lights, which is in green shade. And the third one would be the cone, which is sensitive to shorter wavelength lights, okay, which is in blue shades. So basically, okay, this is the eye, and as you know, this is the retina layer, the most inner layer in the eye. And if you can see, okay, this blue, red, and green is the photoreceptor layer in the retina, which contains the cone. So these are the cones which is responsible for sensitivity to lights or to the wavelength. Okay. So now we move on to the brain interpretation. So these cones in the retina layer, once they already receive the lights, they will process the information, okay, based on the wavelength, and then they will actually form signals. They package all the information, okay, into in the form of signals. And the signals from these cones are sent to the brain, where they are processed to create the perception of color. So that means this bundle of information will be transferred to the brain, and then the brain will actually, will process it again to understand the information that is received from the cone signal, and then they're able to tell you that, okay, this one is containing information about longer wavelength light. So the color that we are seeing is more to this reddish color. So particularly since longer wavelength have a range, so this brain already have the, the idea of, if let's say it is 700, then it should be in this color. 710, then it will be different color. So once they already receive that, then they will refine the information, and based on that, we will be able to perceive the color. Although the process, when I explain, it looks like a very long process, but then all this is actually happening all the time. If let's say you are awake for 24 hours, you will be actually able to perceive color. So just imagine how fast the process is actually happening. But when I'm explaining to you one by one, but it is like a not even a split second, even more faster. Okay, the process happens. So the brain combines the input from the different cones to produce the full range of colors we see. Okay, so when the cones send the information, they will send, like, the red cone will send one set of information, and then the blue cone will send one set of information, and then the green cones will send one set of information. But at the end, when it reaches the brain, obviously the brain will actually interpret the red one, green one, and blue one, and then it will have like one more complex process where it has to actually combine all these information together and give us like a wholesome perception. So that's why when you look at like a one colorful thing in front of you or anything in front of you, it's not like only stands with one color, it stands with different colors. So all this information will send separately by the cones, but will be combined by the brains for you to see to perceive the color in the full range. So that will be the how the brain interprets the information that is received by the cones. Okay, change in wavelength and perception. Okay, when the wavelength of light changes, the color we perceive also changes. So as I mentioned earlier, so if you have a red light around 700 nanometer and you change its wavelength to 500 meter, the light will appear green instead of red. Okay, okay, if let's say we have like a machine, okay, so when we have this machine, so there is one light that coming out from the machine and we have a filter, okay, where we can change the wavelength of light that actually passes through the hole for us to see. So if you have actually set the wavelength of the light that you're looking at as 700 nanometer, you will probably see a reddish color. So when you actually move the leveling from 700 nanometer to 500 nanometer, you will be able to see that when you change from 700 to 500 nanometer, you will see that when it reaches at 500 nanometer, you will see a green shade. Okay, so when the wavelength of light is changed, okay, the color is also changed accordingly. Okay, so that is the connection between the wavelength of light and color vision. Okay, now we move on to the cone photopigments. Okay, so we're going to look in detail what is actually in this, this structure here. Okay, so these molecules are essential for color vision and are found in the cone cells of the retina. And these cone photopigments have a, is even divided further. So we're going to look at what is inside the cone. Okay, so we know that we have retina layer, and in that retina layer, we have this thing, okay, which is the cone layer. So now we're going to look at inner part of the cone layer. Okay, so this is how it looks like when you actually, look into the cones. Okay, so this is the structure. Okay, so basically there are two important structure in this cone photoreceptor. So one is chromophore, another one is opsin. Okay, so chromophore and opsin. So first we're going to look at chromophore. So chromophore is identical for all cone photopigments. The red, green, and blue cones, okay, or the long, medium, and short wavelength sensitive cones. They three have, okay, if let's say it comes from, this red, green, and blue cones, they all have the same chromophore. So they don't differentiate they are not different from one to another. Okay, they all have the same chromophore, the same structure in all the cones. Okay, and it is found in the retina. It is made up of aldehyde derivative of retinol, okay, which is actually a vitamin A. Okay, so this is the reason why it is always recommended for patients to actually consume vitamin A for the eye. So this is the reason why. With the consumption of any vegetables or fruits which contains vitamin A, that will actually helps to maintain the chromophore in the eye. Okay, and the light quanta, light that are observed by the chromophore, initiating the series of events leading to the vision. So once the light enters in the eye and then absorbed by the photoreceptor, okay, the first place that it will enter in would be chromophore. So once it hits the chromophore, and then the initial events, okay, initial process of color vision or vision will start. Then only it goes to the opsin. Okay, So opsin is another structure. It is visually inert chain of amino acids. Okay, it is located, okay, in interlace. It is actually, opsin is interlaced into the dismembrance of the cone's outer segment. Okay, so it is actually here, can be found here, the opsins. And what is the function of this opsin is, it determines the photopigment's absorption characteristic. Okay, how it can actually determine that? Because opsin is not like chromophore. Okay, it is different from one type of cone to another one. Okay, that means the opsin for rate the structure, okay, of opsin in red cone is different from green and it is different from blue. So they are not the same. So that is the reason why, okay, opsin determines the photopigment's absorption characteristic. That means if let's say it is a long wavelength light that enters into the eye, what will happen is it will not absorbed by the green and blue cones. It will be absorbed by the red cones only, because they have an opsin which is actually works like a magnet, okay, which get attracted to only the red lights or the longer wavelength lights. While this green or the medium wavelength sensitive cones, their opsin, okay, is only attracted to the medium wavelength lights or the green shade lights. Okay, so that is meant by the it determines the photopigment's absorption characteristic. So each class of cones has different opsin, which is responsible for detecting short, medium, and wavelength and long wavelength of light respectively. Okay, now we move on to the genetic basis. Okay, so the genes for opsins have been identified. Okay, for the medium and long wavelength cones, or the red and green wavelength sensitive cones. Okay, the genes are located on the X chromosome, which is our sex chromosome. Okay, which determines our gender and all that. Okay, so while these short cones, okay, it is located in chromosome 7. Okay, and when we're talking about photoreceptor, we have cones and rods. Cones are the ones sensitive to colors, while these rods are at the peripheral. Okay, cones are concentrated at the center, rods are concentrated at the peripheral visual field, which actually help us while we are moving around. Okay, while cones is actually, important for color vision perception and also detail looking. So these rods are actually the genes, their genes is located at chromosome 3. Okay, so only these M and L cones, the medium and long, or the green and also the red cones, are the one that is actually carried from one generation to another generation. Okay, so if let's say you have, one particular, uniqueness in your green and red cones, that will be transferred to your kids also. Okay, or to your, future generations. Okay, so opsin genes are homologous to the rhodopsin gene, indicating a common evolutionary ancestor. So as I mentioned, they are carried from one parent to offspring and other generation. Okay, so whatever that you have, it is carried from your ancestors. Okay, while these short cones or the blue cones and also rhodopsins, they are not actually, carried from one to another. Okay, so that's the next one is the mechanism of vision. So this is what I have been explaining just now. Okay, so what happens is we're going to look at the process. Okay, first, photon absorption or light absorption. So light enters the eye and reaches the retina. Okay, it goes into the eye, it goes to the retina, and then once it falls on the retina, activation happens. The lights absorbed by the chromophore, okay, the first structure in the cones. okay, then it causes some changes to happen in the cone. So that will actually induce the opsin. Okay, so this signal transduction change triggers a series of biochemical events in the opsin. Okay, so that means once the light enters, fall on the retina will actually make some absorption of light, okay, through chromophore, the structure in the cone, and then it will activate the cone. So once it gets activated, then what will happen is the opsin will be, kickstart. So this once, the opsin is kickstart, so it will actually open its, attracting, characteristic where it will absorb all the lights that is related to its characteristic. If let's say it's red cone, then it will absorb the red colors, okay, or the red shades or the longer wavelength. Okay, while the green one, it will do its sub also. So when this happens, what will, after that, this information that is received from chromophore transfer to opsin, and then this opsin will produce signal. Okay, so the signal that is produced by the opsin will be sent to the, will be converted to impulses. Okay, it will be bundled up and then changed to impulses, and this impulse will be sent to the brain, and then brain will do some processing, and then it will actually lead to the perception of light and colors for human. Thank you.